The sample required for a parvovirus PCR is a fecal sample or possibly a gut contents. As well as the fecal sample from the patient, we require negative extract control and a positive extract control. The extract controls are there to show that in the negative sense that the assay is not contaminated, and for the positive that the assay extraction process has actually worked correctly. To clean up our sample, we use a mixture called kaolin, which is in salt water to make it into a solution. So the kaolin itself is a light clay, and it helps us pellet down the solid matter from the fecal sample. In addition to the kaolin, we use a compound called DTT. The DTT's purpose is to break down disulfide bonds within proteins. So this helps us remove large proteins which may be inhibitory to the PCR that come from the fecal sample. Things like hemoglobin, which are large proteins that may cause an inhibition in the actual PCR process. So here we have our aliquoted kaolin and DTT mixture. One 15ml tube for each sample. To this kaolin and DTT mixture, we're going to add a pea-sized amount of faeces from each respective sample. Each sample is mixed vigorously on the vortex in order that the faeces and the kaolin mix. The samples are then incubated at 37 degrees for 15 minutes. The resultant mixture is then poured off into a 2ml tube in order that it can then be centrifuged. During this centrifugation, the kaolin and the solid matter from the fecal sample will bind and be pulled down by gravity, the centrifugal force, to the bottom of the tube, and it will allow the sample to split into two separate layers. So we'll have the kaolin and fecal matter on the bottom layer and a nice clean supernatant, the salt water, bacteria, viral particles, etc. from the sample on the supernatant. The next step is to perform some dilutions. The dilutions are to help remove further inhibitory factors that may be within the fecal sample. So we have 95 microlitres of water in each of these tubes, one for a negative extract control, one for a positive extract control, and one for a sample. In addition, we've added a positive assay control. This is a known positive DNA for parvovirus. And again, in addition to that, we're going to add a spike. The spike is a test using our fecal sample from the patient and a dilution in positive control. So for the sample proper, we've got a dilution in water. For this one, it's a dilution in positive control. The reason we're doing that is to try and show that there is no inhibition being caused by the fecal sample. The positive control should give a band exactly equal to our spike for our sample. The reason for this is there is no difference other than the fecal sample being added to the spike, therefore you should get a band of equal intensity for both the positive and for the spike. So here we're diluting our supernatant in the respective samples, and again the only difference between the spike for the sample and the assay positive control is that we've added that small amount of our fecal supernatant to the spike sample. Once the samples have been added, give them a quick vortex to make sure they're mixed, and then the samples are then all boiled for 10 minutes, and that's to rupture any virus particles that may remain and release the DNA into the liquid. Once the tubes have been boiled and cooled, the sample is then ready to be loaded into the PCR reaction tubes. So here we have the reaction tubes which have already been previously made and tested. They're made in batches to make sure that the mixture is consistent. We add our 5 microliter sample to each respective tube. So we have tubes for all our controls and tubes for all our samples and spikes. Once all the samples have been loaded, the tubes are then closed and then centrifuged to make sure that all the liquid is in the bottom of the tube where the actual thermal cycling is going to occur within the PCR machine. So the samples are loaded into the thermal cycler, also known as a PCR machine. The program is started. The program for Parvo PCR will take 2 hours and 21 minutes to run to completion. Once the program is complete, the samples are then removed from the thermal cycler and the samples are run on a gel electrophoresis in order to visualise the bands that are produced from the DNA that may or may not be within your samples. Once the electrophoresis is completed, the gel is visualised using a UV camera. The gel has been stained with lithium bromide, which is excited by UV light, and it allows you to see the bands under the UV light of the camera.